My dear students of 12th class, and today we will continue with our replication, that is the DNA replication, and our topic is semi-conservative mode of DNA replication. And this is the view of the DNA getting replicated. DNA, as you know, is capable of producing its own replica. That means this DNA has the capability of producing the carbon copy of which one of the strands, as you know that the DNA is a double helical, it has a double helical structure, and one of the helices remains conservative, and only one is newly synthesized, and that we know by the semi-conservative mode of DNA replication. And now this was proposed by Watson and Crick when they first described the double helical structure of the DNA. And while proposing the double helical structure for the DNA, it was Watson and Crick. They had immediately proposed a scheme for replication of the DNA. To quote their original statement, that is as follows. The statement Watson and Crick quoted in 1953 when they gave away the double helical structure of the DNA model is, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." These are the words said by Watson and Crick when they proposed the double helical structure of the DNA. And the scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. That means the original double helix, the two strands of that get separated during the synthesis of the new DNA, and the complementary strands are then synthesized from each old strand. And in this way, after the completion of the replication, each DNA molecule would have one parental and one newly synthesized strand. And this is the central theme of our topic today, that after the completion of the replication, each DNA molecule would have one parental and one newly synthesized strand, and this scheme was termed as semi-conservative DNA replication. And now the proof about the semi-conservative mode of the replication, that is the experimental proof, and it was proposed by Michelson and Starr. They performed an experiment through which they proved that this DNA replication is actually semi-conservative in nature. And it was now proven that DNA replicates semi-conservatively, and it was shown first in E. coli, Escherichia coli, and subsequently in the higher organisms such as plants and human cells as well. And it was Matthew Michelson and Franklin Starr who performed the experiment in 1958 that proved that this DNA replication is semi-conservative in nature. And they grew E. coli in a medium containing NH4Cl, that is ammonium chloride, in which the nitrogen part component was the heavy isotope of nitrogen, that's N15, as the only nitrogen source for many generations. And the result was that N15, that is a heavy isotope of the nitrogen, N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation because of the heavier nature, because it was heavy than the normal one. And in a cesium chloride, density gradient, the centrifuge. That means this N15 and the N14 got separated on the basis of their densities. And then they transferred the cells into a medium with normal NH4 cell for the N14, that is normal nitrogen, and took samples at various definite and time intervals. As the cells multiplied and extracted the DNA that remained as double-stranded helices, the various samples were separated independently on cesium chloride gradients to measure the densities of DNA. And now, the centrifugation you must be knowing. It is here, and this is the first, that is the loaded DNA with, on the left hand side, that is the DNA with N15, nitrogen 15, heavier isotope of nitrogen, and it got then after the replication, the two newly synthesized DNA in the normal media, they got one strand of N15 and another with N14, and finally it was seen that this N15 got reduced to 1/4, that is only two strands you can see are red in color with a heavier nitrogen, and the remaining six are with the normal N14 nitrogen. This is very simple to understand, and E. coli has the capability of reproducing fast. It reproduces in just 20 minutes. It takes only 20 minutes to get reproduced, and so this experimentation does not take much time. So the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that's after 20 minutes, E. coli divides in 20 minutes. This you have to keep in mind. Had a hybrid or intermediate density DNA extracted from the culture after another generation, that is after 40 minutes, was composed of equal amounts of the hybrid DNA and of the light DNA. And if E. coli was allowed to grow for 80 minutes, then what would be the proportion of the light and the hybrid? It will get reduced to further it will get reduced, and very similar experiments involving use of the radioactive thymidine to detect distribution of the newly synthesized DNA in the chromosomes was performed on Vishya fava, that is fava beans, by Taylor and his colleagues in 1958, and the results were the same. That further strengthens this proof that the DNA replicates in a semi-conservative manner. These experiments proved that the DNA in the chromosomes also replicate semi-conservatively. Thanks. Have a nice day.